Okay, thank you very much for the invitation, the kind invitation, and having us over and talk about our work. I just had a massive sandwich, so I feel like this is a perfect time to give a lunchtime talk. If you feel like during this talk you're not really following anymore, then please let me know, and either I can increase the pace or decrease the pace. If you have any questions in between, then just raise your hands. The structure that I want to follow for my talk is I have these three sections, three things that I want to go into. Any of these should not be taking longer than 15 minutes by themselves, and I plan to take a break after each one of them, so there is going to be a little bit of time to ask questions to these specific aspects. I have never before given this talk in this way before. So it's going to be a bit interesting to see how everything works out. There could be too many slides in there. There could be too few. I think I'd be happy with too few slides because that gives us more time for discussion and more time for questions. If there's too many in there, then I might have to skip one or the other thing. But Raquel has planned these fantastic coffee breaks, so please let's continue the discussion there if we don't have the time during the talk. So it's a bit interesting to revisit this topic because I haven't actually been using frequency tagging over the past 3 or 4 years that much. But it's always a topic that kind of like haunts me for the reasons that Raquel has already mentioned. So there's this, I don't know whether I want to call it a debate, but this ongoing question, this ongoing issue of how it actually relates. Frequency tagging has been around for a while. How does it relate to this entrainment topic, which is the third and last thing that I'm going to talk about? Before we get into that, I'm going to give you some, a little bit of groundwork in terms of frequency tagging. Where does it come from? Maybe stuff you already know, but still interesting to know and to get into the topic. Then there is this bit in the middle of the talk, methodological considerations, that is maybe the most, in a way, maybe most boring part, so that's why I put it in the middle, so we have something to look forward to after that bit. And then I'll give you my perspective on entrainment right after. I've actually started, the reason why I'm here talking about frequency tagging in the first place is because I have done a lot of frequency tagging in my PhD and also during postdoctoral times and came across a lot of people that I was able to discuss this topic with, and I have some insights into methodological developments and so on. And I hope that becomes clear in this talk. And if not, then again, just please let me know if I need to go into something in more detail. In this talk, I think most of the slides are basically just, showcasing one particular aspect of frequency tagging research or a methodological approach or something like that. So it's basically just spotlights on interesting stuff. And we don't really have the time to go into any of these in particular. But there's always more sources about that, and if you're interested in any of these particular things, then let me know after or in your questions. Okay, so that's enough of a disclaimer or preamble, if you wish. And I do have this thing here, so I can actually walk around. It's great. Put myself here and show you stuff. So let's have a look at this first part of the talk, where we look into the origins of frequency tagging, and I think you're all familiar with how it started. Frequency tagging is, of course, very closely related to the progress that we have made over the past century in terms of describing human brain activity with electrical recordings, and Hans Berger was the first to do that, as you all know, with these funky electrodes from our perspective today. One on the forehead, one on the back of the head, and then he was the first to describe these fluctuating voltage changes in the human EEG. And in his case, this is here the very typical case where on this sort of like paper strip that the pencil was scribing on, he instructed the participant to close their eyes, and then you can see this, what we now know as the alpha rhythm, go away after. So the first thing that you actually saw in the EEG, except for, on top of the motion artifacts of the participants, probably was something rhythmic, right? Just as a side note, maybe for the history buffs, is that it's really quite impressive that back then, with these techniques, he managed to record over 300 EEGs, and he published 14 papers. And interesting from our perspective today, all of those 14 publications have the same title, so I don't know whether you can pull that off nowadays, but that's interesting. Okay, so the point why I'm showing you this is, of course, the oscillatory part of that, right? And it only took people a few years, Adrian Matthews in this case, to come up with a really ingenious idea of saying, okay, so the brain produces these oscillations by itself, spontaneously, we just have to record EEG, but as an experimenter, we want to make sure that we have some control over that. So can we devise of a situation where we can produce these oscillations? And they came up with this contraption here, where they put participants in this head box, basically, facing this shade, this opal shade, and then had these lamps produce rhythms for the participants, so quite a heavy type of stimulation, and were then recording the produced brain responses and analyzing it. I'm not going to go into detail what they found, just so you know. The idea was around very early on. After first describing brain rhythms, Adrian and Matthews were already driving brain rhythms also in the alpha frequency range. And they back then they coined this term of photic driving, so this might creep in at times, and now you know what I'm talking about. So this is the first kind of like way or term to describe frequency tagging back then, right? If we move forward a little bit, it's interesting to look at the technical sophistication that people went at this. There's maybe one thing that you need to look into in this picture here, that is the work of Gray Walter in the 50s and 60s, where he, with lots of hardware, set up this toposcope lab and had participants come in. If you look at this closely, you see the participant is lying here with their eyes closed, actually, and then had this strobe light put in front of them. You can imagine what kind of like stimulation that must have been for the participant to be flashed with this kind of light. And then they had their EEG recorded, and that went through these amplifiers here. And you also have the paper machine here for recording the EEG. And the most interesting part actually is this device back here. So they built this apparatus that allowed them to take the signal that they recorded also from quite a high number of electrodes for the times back then, and look into the phase, the relative phase of the rhythms that they recorded from the scalp here, right? So I'm assuming that you're all relatively knowledgeable when it comes to oscillations. You know, oscillations come at a certain phase, and if you drive an oscillation and record it with that, then at different points of the scalp, you will record this oscillation with phase shifts relative to each other. And they were looking into that and trying to devise from these phase shifts how these waves actually travel across the scalp, okay, or within the brain, inferring that from what's happening on the scalp. And they did that online, and that back in the 50s and 60s. Okay, so that's just a very short kind of like foray into the history of that, and how long photic driving has already been around, and how it also helped us drive the technical advance of EEG. Some of the systems that we use today go back to what they did in the 50s and 60s in that way. If you want to know more about that, a dear colleague of mine, Keitel, published a nice paper about this, what she called EEG archaeology, where she looks into just this period and also with a focus on what people looked into in terms of alpha and frequency tagging or photic driving. So that's definitely, it's a short read and a worthwhile read. Have a look at that if you want to know more about what I just told you. If you go on a little bit, then in the 60s and 80s, the basic frequency tagging or photic driving work is probably best described as technical groundwork, where it was much more, people looked much more into the neurophysiology of things, so kind of like conduction times and neurons. If you want to read up on that, then this is the Bible, essentially, this book by David Reagan, so they've done a lot of work in that. I think especially on the neuronal level, not really with human subjects. They did that too, but essentially they were looking into the neurophysiology of these signals. And then I think what really started the area of research that I'm talking about today mostly is what set this off is this 1996 study by Morgan, Hansen, and Hilliard, where they found out that they can use frequency-tagged brain responses, stimulus-driven brain responses, called steady-state responses, to look into a cognitive modulation. So of course, for us cognitive neuroscientists, that's a fantastic thing to have. You have a driven brain response and you can show that it somehow modulates if you manipulate something like attention. Okay? And that's what set off a spree of studies. So there's been an increase, and this is basically what I call the modern frequency paradigm, frequency tagging paradigm here, what kind of like started with this particular study, where the idea is always you have some sort of like visual stimulus that can be as easy as a light being switched on and off. It can also be a color change, anything, any physical change of a stimulus over time, periodic change that then drives the brain signal at the same frequency as the stimulus. Right? So that's always the idea. You have a stimulus that is going on at 10 hertz. You produce a brain response that also has a frequency of 10 hertz. And what I show you here is already one of the advantages of frequency tagging. You cannot only just use one stimulus, but you can use several stimuli. As long as you use different frequencies, you can later distinguish the responses to these different stimuli that you show on the screen by just, separating them in the spectral domain. So this stimulus has a 10 hertz response, the stimulus maybe has a 7 hertz response, and if you analyze your data spectrally, your EEG data, then you will be able to look at those. And if you do an intentional manipulation, participants look at one stimulus versus the other one, then you will see an effect of the intentional manipulation. We'll look into that later on. But kind of like flavors of that have been used in what I consider to be the area of frequency tagging research that I'm most familiar with. And let's have a look at what this looks like today. So I called it recent past or recent present. I think we're going to hear from Noor later on what the actual current frequency tagging research looks like. So this is just what has happened in the last 20 years. And I've talked about how attention kind of like kicked off the frequency tagging thing. We also have other things that people have looked at, for example, the effect of emotional expressions, emotional pictures, emotional context, content and picture materials, and its effect on early sensory processing. So the thing that we look into with frequency tagging, there is a prominent line of research looking into how we process faces using frequency tagging, figure ground segmentation, multiple object tracking, and then has been something going on here in Ghent as well. So you have Dirk Talsma here, who has looked into multisensory processing using frequency tagging. So you can actually ask him if you have any questions about frequency tagging. You don't need us. And other concepts as well. So working memory, for example, aging, and also sleep. So there's an interesting study using visual frequency tagging on sleeping participants. I was never sure how they can actually sleep while being stimulated, but it seems like it's working. So through closed eyelids, and it allows sort of like an assessment of sleep state as well to look into the response there. Okay, so this is where we are just now. And I quickly want to go over the advantages of frequency tagging. That one of them I have already alluded to. This is an example of, this is a relatively extreme case of a study that we did in Leipzig, where we had an array of 6 photodiodes, and all of them were flickering at different frequencies, from 9, 10, up to 14 hertz. So all of these on at the same time for several seconds on the screen. I'm not going to go into the design of the experiment too much. Suffice to say that this was also about attentional allocation. We wanted to see whether participants can split their spotlight of attention. And the good thing, another advantage, on top of you are able to track the processing of several stimuli at a time, is that you have rather constant stimulation. So apart from the fact that it's flickering, it's going to be there for several seconds. So you have, if you wish, stimulus constancy, right? So something more closer to our real world when you compare it to the classical ERP paradigm where you just flash something on the screen and then you measure the response to that. So you have something, as long as the stimulus is on and flickering, you get a steady state response, so you can play that for several seconds. This is also what I told you earlier. If you have several stimuli on the screen and all flicker at different frequencies, when you do a spectral decomposition of your EEG later on while participants were looking at that, you get these peaks in the spectrum at the stimulation frequencies. So we were able to separate the processing of all those stimuli and kind of like look into the attentional modulation. Like I said, I'm not going to go into detail with that. Tracking of multiple stimuli. And yeah, of course, the biggest advantage, and that's why people are using it, if you have some sort of like cognitive process that you think should have an impact on early sensory processing, then you can manipulate that in your experiment and see what kind of effect it has on the steady state amplitude. The typical effect of spatial attention and feature-based attention, for example, is that if you attend to a frequency tagged stimulus, the amplitude is going to go up. And the same holds for other manipulations. This is also a really nice example that plays into sort of like a different direction in terms of an advantage of the technique. Not only can you look into, so here in this case, I'm kind of like looking into a 2 or 3 second window of stimulus presentation. So I get kind of like a statistic, an average statistic of the attentional manipulation of stimuli over those 2 seconds, but I lose all the time information. What I can do instead is do a time frequency analysis with my steady state response and see how it modulates over time. Okay? Here's an interesting experiment where they had the typical phase-vase illusion. Hallucination? No. Illusion, thank you. Okay, so this phase-vase thing, I should have put the picture here, but I think you all know what this looks like. Where you can either see an ambiguous vase or phases in this picture, and they frequency tagged the vase part of the stimulus at 12 Hertz and the phase part at 15 Hertz, and then had participants look at this and press a button every time they felt like the perception of those two things switched. And in this very representative example of the data, I'm sure, you can see that it actually tracks the switch, right? So you have a relatively higher 12 Hertz amplitude in your response, as long as the participant sees the vase relative to the 15 Hertz signal. And once the perception switches to the phase, you get a higher signal in this frequency band. Okay, so that's quite an interesting and sort of like easy to explain example of how you can use the temporal information. People have also looked into times, so also the timing, the time course of any switch, any attentional switches can be interesting in your steady state response. You just have to use frequencies that are fast enough in order to be able to do that. And one last thing that I quickly wanted to mention is that, of course, we can use that for basic neuroscience to learn something about cognitive processes, but people have also been looking to applications of frequency tagging. And one nice example is that it's used in a way to, kind of like control something with your mind, like this computer game here. This is an early study by Layla and colleagues, where they had this monster balancing on a slack line, and was to, covertly attend to the left or to the right checkerboard, flickering at different frequencies. And with wherever you attended, you had to balance this monster on the slack line to kind of like safely, just by allocating your attention to one of the two frequency tagged stimuli. This result and other results have led to of frequency tagging in some different brain-computer interfaces, for example, speller brain-computer interfaces, where you have a display of letters, tagging frequencies across. I don't know, I'm not really up to date on how well that works, and navigation is really close to this particular example here, where you can either control something that's around yourself through tagging. Good. I think telling you frequency tagging idea itself is really almost as old as the EEG. We see these simulations, can we do them simulation? Then the times that we talk about today from what happened in the 90s, and frequency tagging as kind of like a neuroimaging technique in its own rights, then that I was able to conclude that there are not too many disadvantages. You'll hear later on in Noor's talk. Okay, that takes before I go, might answer a few questions or something that I've shown you that here.
So I assume that the most strong C is probably on posterior electrodes. Could see
You mean in cross the scope? Yeah, that's going to come up, but brief response, and the signal is strong enough to be able to record it. But you're right, occipital posterior parietal electrodes should usually be strongest if you use visual stimulation, of course. Yeah. Okay. Good. Gonna go and move on. And earlier advertised as the most boring of this talk, the stimulation parameters. So if you want to do frequency tagging, this is one thing that you need to pull this off. And of course, visual stuff. The first thing that we are going to talk about is what do you need for your frequency to see a response in your EEG. Of course, I think that without saying, the most important thing is so the space. Of course, at some point is going to be too strong and run into a situation with intensity that can lead to an epileptic seizure. So don't. But as long as I have been doing frequency experiments, I never had this situation. And then again, my stimulation was always relatively low intensity. Here's an frequency. So the next parameter that you should think about is so at which frequency stimuli. And the classical example earlier have all used frequencies in the lower frequency range, so maybe up to 30 Hertz. In the literature, visual frequency tagging in this range, and in terms of power and the signal-to-noise ratio that you get, that seems to have been a good choice, because you can see this kind of like it goes up until roughly Hertz, and then it goes up until these higher frequencies. So you always get a response to your signal, but the signal-to-noise ratio will be much lower at higher frequencies. And here, interestingly, the frequency where usually you start losing the sense of something flickering on the screen, moving into a more permanent stimulus. So this frequency here. Don't know what that means, to be honest with you, but it's interesting. That I want to go into detail on is the waveform of the stimulation. So again, this in its space that you can cover. Typically, people have just used these square wave stimulations where you switch something on for several amount of time and then you switch it off. Usually, the on and off period is 50-50, but you don't need to do that. You can go for a more permanent stimulus if you go 90% on, 10% off. Problem is, your signal will be weaker after if you do that. So, so this is the square wave. You can also go with different forms of stimulation. In visual frequency tagging, it's a bit difficult to do that with the computer. But if you have photodiodes, for example, you can feed them like a sine wave, so you have a more smooth way of stimulating. Into how these parameters actually affect the signal that you're driving. There is one that I cite here. Are these slides available after? So, yes, okay. Okay, good. So I can just point to that and say, look at this. There's one study that shows how to interpolate frequencies that you don't naturally have on a given computer screen using approaching a more sinusoidal function. And apparently that in that case didn't really do much in terms of the amplitude of the steady state response. So maybe you're safe there. But it's certainly something to look into if for some reason your experiment necessitates a deviation from the standard square wave on-off thing. Yeah, and there are other things to think about, especially if you're going with kind of like a different visual feature. Usually it's like a luminance manipulation. If it's a color manipulation or a contrast manipulation, the story might be entirely different. So like I said, there are not too many studies around, but there are some that look into the difference and what you actually change in your stimulus and how it affects the signal that you drive. And that's something that you always need to keep in mind. Another thing is that, so I'm talking about the visual part of the whole story for most of my talk, and that's why it's easy to say, look, you can do that in vision. But frequency tagging has also been used in sound stimuli. You've probably know or probably read about auditory steady state responses, frequency following responses. Usually what people do there, so they can do the very same thing. You have a stimulus, an auditory stimulus, and you modulate it at a certain rate, and that produces responses at those rates. Usually the frequencies are slightly higher, so it seems like between 30 and 40 hertz is a nice frequency range to drive strong responses in the auditory domain, which might have something to do with the properties of the auditory system. And you can also do that in touch, so tactile stimulation. I don't know, I hope you can see that to some extent. You can also use either sort of like mechanical flutter on the fingers or any other body part for that matter to drive a tactile steady state response. People have, I think, also used short burst laser stimulation to drive different receptors. And it works too, maybe at a slower rate. But again, here, if you use mechanical stimulation, then you might want to go into the 20 hertz range to get the optimal response out of your data. Again, not necessarily something that has been tested parametrically that well. Okay, there are also some exotic cases where people have driven I might consider it frequency tagging. The people who have done it might not, but the principle is always the same, that you vary something, some sensory input in a periodic fashion, and then you measure the brain response to it using spectral decomposition. In that case, for example, participants, I think they stood on a board and that was tilted at a rate of 1 hertz or something like that, or could also be that this is an electrical stimulation that they have used in this case. I don't really remember the details. So this definitely plays into the direction of vestibular input. So you can drive a steady state response by changing vestibular input periodically. And you can also give olfactory input by, just shooting a few molecules at the nose of your participants at a certain rate. And that gives you a very, very slow olfactory steady state response. This already goes into a direction that I might talk about later on, but maybe I can mention it here already to prime it a little bit. So what people are doing usually with the regular stimulation sort of like lends itself to later using spectral decomposition, right? So you know something is going on rhythmically, so that means that you can look at your EEG data using a very similar approach, and then you get a peak in your spectrum. One thing that is going to be interesting later on is that this shouldn't be taken as a sign that there is something oscillatory going on in the signal. It just means that there is something periodic in the signal, okay? And I hope the difference becomes clear later on as well. I just wanted to mention it here because I felt it is worth it. Okay, so this is the kind of like the part about the stimuli. What you should keep in mind, you can use several different modalities, but you need to make sure that you get your stimulus parameters right if you want to do frequency tagging. And the next bit is, once you've done your experiment and you have your data, what can you do to the data to optimally extract the spectral signature from the data? It's just a couple of things that I like to do to the data. The first one is actually both go in the same direction, both go into the spatial filtering direction. And there is an argument that can be made, and this is something that might be more true for visual steady-state responses, that you should be looking into scalp current densities, the scalp current density measure of EEG rather than the voltage itself. You can compute that from the voltage. I think if you use field trip for data analysis, for example, they have that nicely built in. So it's a one-liner in MATLAB to convert your voltage into scalp current densities. And what that does is it usually focuses the scalp projection of the steady-state response generated by the stimulus. So I have an example for you here, unfortunately not from the same data set, but same frequencies and roughly similar stimulation. So we have lateralized stimulation. We have a stimulus left and a stimulus right. One flickering at 10 Hertz and the other one at 14 Hertz. And if you just look at voltage topographies, you see that it looks a bit all over the place, right? So usually you would expect you have a stimulus in your left visual field, you have a nice blob on the right hemisphere, but that's not what it looks like. That has something to do with the fact how the generators of the steady-state response in early visual cortex are sort of like oriented and then project to the scalp. So you see something all over the place. You even see something frontally here. That's why I responded to the question earlier with you can actually also record the steady-state response here if you just go by the voltage measure. What you can do though is if you convert that into the scalp current density, you see that you have these nicely localized maxima of the response. So what it helps with, so it's kind of like a poor man's source localization, if you wish, but just on the scalp. It kind of like focuses the sort of like the scalp projection of the steady-state response closer to the actual source of where it's coming from, where it's kind of like coming out of your brain and projects onto the scalp. So I can recommend this bit, and it helps a lot with an issue that sometimes plagues you when you do this kind of analysis, that is picking the electrodes that you want to throw into your statistical analysis, for example. Can be a bit tricky if you have that. It's easier if you do that. And then there's an even better approach that is a bit more recent. It's called rhythmic entrainment source separation. I don't like the name, but I haven't invented the algorithm, so it's still great, and it's based on this generalized eigenvalue decomposition. It's always nice to say it, but I'm not a mathematician, so I don't know necessarily exactly how it works. But what it does is completely alleviates any necessity to pick a particular set of electrodes for your analysis, because you just define the frequency, your stimulation frequency, as the one that you want to look at, and then it optimally weights all the electrodes that you have on your head and gives you back one component that represents this signal optimally. Here's an example. So if you just pick OZ as the electrode that you want to analyze, you get this particular signal-to-noise ratio. And if you apply this algorithm to give you a component that best represents the signal, then you get a much higher signal-to-noise ratio, so almost like a threefold increase in signal-to-noise ratio. It's a bit tricky to apply this, but we have done this in one project, and again on the slides you have a link in for some inspiration of how to do it. In this case it's a field trip, and the original work is from Cohen and Gulbinaite, who have made the code available at the time, but I think it's not available on the website anymore where I first got it. So you can find a version of it here, and I think it's also been implemented in a toolbox that I don't know, but maybe you do, the MEEG kit, which has this fully implemented, where you don't really need to do much.
It's a relatively new paper by Mike about this, and it's a tutorial paper.
Okay, so perfect. So it's probably a good idea to link that before sending it out. Nice. Good. Okay. So this is getting the signal out of the data. and these two things might help you, especially if you are looking into visual steady-state responses. And then the last thing that I wanted to go over real quick is how you actually quantify this signal in your data. And classically, what people have been doing is this here, which is called evoked power. And it basically just means that you calculate for the trial that you have in your experiment, you calculate the ERP. So you kind of like average all the trials, and then you apply to that a spectral decomposition frequency transform, and that gives you this picture here, where you have, in my case here, a stimulus that flickers at 10 hertz and another stimulus that flickers at 12 hertz, again, left, right. That works well. You also get this kind of like difference between the two frequencies, which is a little bit an artifact of this approach, and this is why I have so for myself decided that this is not really my favorite approach anymore, because it also confounds a little bit two things, and that is the actual power. So you think you get power in your estimate here, but it is actually also a measure of how well aligned the oscillation is across trials. So what a way to get rid of the power thing and only look into the alignment across trials would be to calculate something called intertrial coherence, which I think you're also familiar with. Then you can actually see that those two responses are roughly similar in terms of how well they align across trials. And the thing that I haven't really talked about here is that you also get a feeling of the attentional modulation, right? So if you attend a stimulus, you get a higher intertrial coherence than when you attend the other stimulus in that particular design. Okay. You can also go for slightly more exotic measures if you want to. I've played around with something that is called the cosine similarity index, and there's a paper on it that I also haven't linked, I just noticed. So another thing to add before we send this around is that you can apply that. And the benefit of this particular measure, which is actually something taken, I think, from image comparisons, but somehow it also works really nicely with spectral data from frequency tagging experiments, is that it is less susceptible to a situation where you have a vastly different number of trials between conditions. So this index here somehow is robust against that. To a certain extent, or certainly to a higher extent than looking into intertrial coherence. So again, if you wanted to apply something like that, then this project that I have advertised earlier also has that, and it's worth looking at. That was the last thing, sorry, there's one more thing. One thing that I've been looking at sort of like as the last step in doing frequency tagging analysis is thinking about why we always use rhythmic stimulation and whether that's not a really super artificial case, especially if you think about visual dynamics as something that we encounter in everyday life. And examples for that are speech stimuli and maybe making hand gestures while I'm speaking. So we know that these play a role in parsing language signals, for example. And it would be interesting to look a little bit further into how our brain processes these dynamics. We know, though, that they are not strictly rhythmic, but only quasi-rhythmic. So what does our brain do with these signals? And in order to do that, I just adopted a very, very basic frequency tagging paradigm again with these stimuli here, these sort of like patches. And in this case, for stimulation, we used not something strictly rhythmic, but something that varied in frequency over time for both of these stimuli within the same frequency range. In the experiment, we tested 4 to 7 hertz, 8 to 13, and I think 14 to 20, kind of like covering the classical theta, alpha, and beta frequency range to see what the brain would be doing with these signals. And just a very quick look at what we found in this case here is that you can actually also extract nicely from this signal what we call the brain stimulus coupling using a measure of spectral cross coherence. This is closely related to intertrial coherence. The only thing that you do is you take the stimulus itself into account and calculate sort of like the correlation with what the stimulus is doing and what the EEG is doing at any time. And that gives you, that sort of like returns the stimulus dynamics as a spectral peak. and you don't see that if you flip the stimulus around, so if you use the reverse version of the stimulus and correlate that with the EEG. So that's a neat way if you want to, so one maybe disadvantage of steady state responses is if you want to separate your stimuli, they have to have different frequencies. This way, as long as you decorrelate these two sequences, you can stay within the same frequency range. That might be an advantage, and another advantage from a more conceptual perspective is that you're closer to natural visual dynamics if you allow your stimulus to vary in rhythm. Okay, so I can quickly summarize this by saying that we do really have a huge parameter space if we look into what actually determines the response that we get out of our brain if we put in a certain frequency tagged stimulus. We can use different modalities than the visual one to do our experiments. And actually, we can also do multisensory paradigms. Dork's experiment that I mentioned earlier, for example, also has a multisensory component. And we've been doing some experiments where you have visual and an auditory stimulus at the same time, or visual and tactile, or auditory and tactile. So all of these things can be done to also study multisensory interactions. And I've also told you then that you can use some different spatial filtering approaches in order to boost your signal once you've done your experiment and you are looking into the data analysis. And we're not necessarily limited to strictly rhythmic stimulation. We can also look into quasi-rhythms within a certain reasonable range of frequencies. Okay, again, that is the part of the end of the second part, and I don't know how I'm doing with time.
And you have like 15, 20 minutes.
Left?
Yeah.
Okay. Okay. Wow. Good.
Including questions.
Including questions. Okay, good. That's good. So any questions now about this part before we move on to the last bit? If not, then I'm just going to go on and.
Sorry, you mentioned in the way you look at the brain response, the fundamental frequency, but you did not mention anything about harmonic frequency.
Mm-hmm.
Would like to have your opinion about that, or
Mm-hmm.
do you include this in your?
That's a very good question. So I haven't put in any slides about that, but you're absolutely right. This is actually a direct consequence of the fact that the brain response that we drive is never sinusoidal and has a certain offset. So if you drive something at x hertz, you always see 2x, 3x, and so on responses in your spectrum too. And my approach so far has been I've only looked at them if they are convenient. But you should actually take all of them into account, because when you think about the Fourier transform as the spectral representation of the signal, only all of them will allow you to reconstruct the whole signal, right? So it kind of like makes sense to look at all of them in concert. We've done some audiovisual experiments where we looked into the effect of multisensory congruence on what it does to the steady state response. And there it was really interesting to look at different harmonics, because they seem to be modulating in different ways depending on the factors. So yeah, should be looking into the harmonics too.
Yes, so I don't know, maybe you responded in the fifth part, but if like this power of this response to this stimulation is different in different individuals, yes? And,
Yes.
For example, in low frequencies we have this individual alpha frequencies. So it's like something that is kind of fingerprint of a specific person, individual. It's this response to specific like frequencies also differ. For example, you could slightly like kind of it could be related to this kind of individual like frequencies. It's like some research on this stuff.
Yeah, so my intuition would be, I don't really have any data on that. First of all, I think you're absolutely right. So there's lots of individual difference in how strong the response is that you can drive. I would say that it's not related per se to the individual alpha frequency or any other individual frequency, but more to the fact that there's also lots of individual variability in the strength of those oscillations. So maybe whatever gives rise to these differences also gives rise to the differences in the steady state response that we can measure. For example, could be something as easy as skull thickness of the participants, or how the generators in visual cortex are oriented in each individual participant, because cortex is always folded slightly differently, and the generators of the steady state response seem to be relatively localized. So if they project in a funny way onto the scalp, you might see a very small signal for one participant, but a really strong one for another one, and that could make a huge difference.
Because one difference could be like just overall a general level of this response. So some individuals could have just lower, but for example, if there is some difference like slightly higher, like one hertz higher frequency, they have the same level of response, but just in higher frequency. Is kind of this phenomenon exist, or it's not? No one discussed this.
Can't really think of anything on the top of my head. I think it would be the easiest thing to look at a given data set to see how strong the response is to the visual stimulation and also to the alpha.
Also from stage, something like parameters.
Yeah.
Some parameters and that to try to predict
Yeah.
or kind of connect this.
Yeah.
But it is not you don't know about.
I think I'm vaguely remembering looking at something like that, and maybe later in the coffee break I'm going to remember. So ask me again. I might remember. Okay. So let's just jump into the last part, which maybe goes into a direction that we were going into anyway, and that is the question about this, big topic, entrainment, right? So maybe just to make sure that everyone knows what we're talking about here from a physical perspective, this is the idea that you have a strong external drive, which you might think you have with a visual frequency text simulation, and then this usually also involves the consequence that you have an internal oscillator that is already oscillating away, like individual alpha frequency, and that gets then sort of like forced into phase with this external oscillator, right? So it's like this old example of two clocks hanging on the wall and swinging their pendulums at different rates and frequencies, and after a while they get in tune. This is what entrainment is, right? And the same idea exists for brain rhythms. The interesting thing is this is kind of like a story told from my perspective, and you might not necessarily agree, but it was really interesting for me during my PhD and as a postdoc later on to kind of being thrust into this world. Because for me, I was always coming from this particular perspective. This is our kind of like design of our experiment. We put something rhythmic into the brain, we get a rhythmic response. We call this frequency tagging, and we just do it to study influences on cognitive processing without having a really strong notion about what this response actually is, right? But then at the same time, so I was going to conferences, as you do, and seeing people talk about this, and the funny thing was people were doing exactly the same thing, but assuming something completely different about the brain response that you're getting. And the idea behind entrainment is that it allows you to interfere with the sensory processing by actually causally modulating brain activity. For example, the alpha rhythm, right? Funny thing is, again, if you look into the experiments, they look very, very similar. So the way that we actually drive these brain rhythms is exactly the same, right? So this is an example from an experiment that I did back in the day where I have left and right stimulation. Looks a little bit different to this, but in the end, it's just two lateralized stimuli flickering at different frequencies in the alpha frequency range. So essentially the same thing, but with very different underlying assumptions. And a friend of mine put these nice monikers on it and said that what we do in a frequency tagging is what you would more dramatically call wiretapping into sensory processing. And if you are sort of like into entrainment, then what you assume is that you're actually hijacking with your stimulation. So frequency tagging as wiretapping or frequency tagging as hijacking of oscillations. And of course, all of this kind of like then leads to the question, what is the actual nature of this response that we're measuring in our data? And if you are a wiretapper, then you can either be on this sort of like level that I've already mentioned. You're kind of like agnostic. You don't really care that much as long as it modulates with your cognitive process. So if you are coming from an experimental psychologist perspective, I think this is a fine way of living. You just want to see how your concept works in the brain, and that's fine. But there's also people who have looked into this a little bit more, and then if you look into how steady-state responses are usually analyzed in frequency tagging paradigms, then I think it usually aligns more with the perspective of an evoked related potential, event-related potential. And there's some data on that. So in the study by Keitel and colleagues in 2010, what they did is they just took ERPs and modeled a steady-state response just by gluing ERPs next to each other, and then kind of like emulating what the steady-state response would look like and what a spectra would look like. And it looked very, very similar to what they actually found when they did the EEG experiment, which gave this whole ERP-based perspective of the steady-state response a good push. But of course, there's another perspective, that of the hijackers, and I should maybe first talk a little bit about the motivation to take on this perspective in the first place. And that is that the response in the eyes of a hijacking perspective is basically identical with some form of an entrained endogenous rhythm. So the brain produces 10 hertz on its own, and with the rhythmic stimulus, we just tap into that, and we start forcing that into phase. And that means that we have, as an experimenter, we have control of the human alpha rhythm, right? So in the spectrum here, that typical 10 hertz peak, this is what we control now with our 10 hertz flicker. And why would you want to do that? That has something to do with the fact that how we think alpha works. So it kind of like is this waxing and waning phase of excitability in our visual cortex, for example, in a very simplified manner here. So at some points, our cortex is very lowly excitable by some external stimulus, and at some other phase, periodically changing, we have high excitability. This is how we think, or one of the ideas that we have about how the alpha rhythm works and has an impact on visual processing. The problem with that is, there's no problem with that just now. I'm going to talk about that later. But what that allows us to do is look into this idea of rhythmic sampling. So the idea that our perception, this really interesting idea that our perception is not really continuous, but we take consecutive snapshots, and our brain then creates the illusion of a continuous perceptual experience. As an experimenter, you would then be able to control exactly when these phases occur, and you can present stimuli there and see whether participants actually detect those stimuli or not. And some experiments have been carried out in that direction and found evidence for that. Also before doing the entrainment. So there was some heyday of looking into the effects of alpha phase on cognition in the late 2010s, early 2010s. Interestingly, we have just last year, we published a special issue in the European Journal of Neuroscience where we tried to collect null findings as to this particular finding, actually. And interestingly enough, we found a lot, and we had a lot of submissions. There seem to be some patterns to that, so look into that if you're interested more in rhythmic sampling. But that would be kind of like leaving the path a little bit for the purpose of this talk. So that's the idea, right? So if you are approaching this from an entrainment perspective, that is what this would buy you. It would buy you control over an intrinsic rhythm, which would then allow you to study this in a much more causal way. Okay? Now, there's just one thing that I think speaks against an explanation of the steady-state response being an entrained response, and you have to bear with me a little bit. I think this is maybe the hardest part of the talk. It's good that I put it at the end of it, so I will have difficulties explaining it, and I hope that you're awake enough to still make it through that part. So here's a situation that you typically encounter in experiments that I call the attentional modulation conundrum. If you look into studies, and there's a huge wealth of studies that have done something like that. You have a participant set up by presenting them with a cue that points to a left or to a right location, and then you kind of like record their EEG while they're waiting for something to appear at that particular location. When you do that and look into the trials after, what you will usually find, looking into alpha power, is that so this is the position that they're looking at. So this electrode here is over a hemisphere that represents that position, okay? So this electrode will then have a higher alpha power when this position is ignored than when it is attended, which has something to do with like how our visual paths cross and how alpha suppression is the kind of like points in the counterintuitive way, right? So it's higher when we ignore a certain position than when we attend one. Something really similar exists in the frequency tagging literature, where you have a situation where participants have to focus on one of two flickering stimuli. They get a cue again that points into the direction of that stimulus, and then they attend that particular stimulus flickering over there, also in order to detect some sort of like a transient target that is being presented on that stimulus. When you look at the response to that stimulus here, you will find, again, this is the response that comes from the contralateral hemisphere. You will find that over time, this is just like a temporal spectral analysis of the data, you will find that the response to the attended stimulus, or when the location is attended, is higher. So you get the complete opposite effect. And this is also true for frequencies in the alpha frequency range or close to the alpha frequency range. What hadn't been done so far is to look into this in the very same data set. And the good thing is that we had a few data sets lying around that had some flicker frequencies in the alpha frequency range. So I thought it might be interesting to look whether we can kind of like find the same pattern in one and the same data set to contrast them directly. And there's a little bit of an extra explanation that I have to make first. That is, I've talked about evoked power first. So we had an experiment where we were driving a 10 Hertz and a 12 Hertz response with two stimuli presented to the left and to the right hemisphere or visual hemifield. As you can see here, you get these responses. They are quite small. The reason for that is that the modulation in the stimuli was relatively low level, or actually quite low level. So the changes on the screen were not drastic, but still we were able to extract these responses. And this is one way to look at the data. Now I'm going to show you the other one. So this is the power evoked by the stimulation, where you average all your trials. And this is what you get when you do a spectral transform of the trial averages of the ERPs, basically. If you do it the other way around, if you just calculate the spectrum for each trial, and then average the spectra, you don't see the stimulation frequencies anymore. You see the strong alpha peak instead, which is also kind of present in this data. So it kind of like pollutes what's going on here, right? The interesting thing, so these two perspectives on the data that give rise either to one response versus the other response is just this minute difference here in MATLAB code, right? So you have some sort of like complex Fourier spectra that you get out of your spectral analysis, and then either you average it first and then take the absolute value, or you take the absolute value first and then take the mean. These two switches lead to vastly different outcomes already. And that's something that you have to keep in mind if you're playing around with that in MATLAB. One thing that I did then is switch this around a little bit because of what I explained to you earlier. Evoked power has this mix of showing you the power of the signal, but also the phase consistency. So for the analysis, we then switched over to intertrial phase coherence, which shows the responses to the stimuli more nicely. And then we looked at those two things. So first, I'm going to show you the ongoing power. So this is where we calculate the spectra first and then average the spectra across trials, which is usually the way alpha power is analyzed. If you do that, you find an effect that is very, very common. This is the typical alpha lateralization. If participants have to attend left versus right, contra versus ipsi, you see that nicely localized in this topography here. And you will find that for the ignored stimulus in that frequency range here, you have higher power and the same for the other side. So that is true for the left and the right stimulus. Like I said, typical lateralization. And alpha power is suppressed contralateral to the attended position. This is what this tells us, right? This is what you see in the spectra. If you then look into the evoked power, what you see is that you actually see in the same data derived from the same analysis, but just with this tiny switch in how you do it, how you represent the data spectrally, you all of a sudden find this actual peak in the data. So if you look at these electrodes here, you see that you have a 10 hertz increase in the attended signal. And here you see that for 12 hertz, the signal is increased if participants attend to it. So this is the typical gain effect that we would expect in steady state effects, in steady state experiments, in frequency taking experiments. And there is no suppression going on, right? So that means that we actually have an attention effect on both of these signatures of neural processing that is completely reversed. And it kind of like has this kind of like dependency on the analysis, which I think might be what is confusing people at times in terms of interpreting the results. And that means that sort of like this is my play on why I don't think that entrainment is happening, because functionally we can dissociate these two signatures and they're doing two opposite things, okay? So alpha shows the typical suppression, the steady state response in the same frequency range shows an increase, so the complete opposite. This is why it doesn't really work for me simply saying that what we're looking at here is entrained alpha. Good. So the summary of this part is that I told you in the beginning how there are these two vastly different assumptions about what we're looking at when we're looking at the frequency tagged response. I think actually we need to resolve this because it can create contradicting perspectives if we are interpreting the data from our experiments. And this is also maybe something I quickly alluded to, how frequency tagging is used in brain-computer interfaces. If we want to use it there, we need to be sure of what we are actually extracting from the brain, and if we're not somehow manipulating brain activity that we shouldn't be tampering with. So if we want to use these applications, we should know. And this is strictly my opinion. If you look into the visual steady state response, I think that we cannot go with an account that says that what we're looking at when we are driving this response is purely an intrinsic rhythm. What I think is possible is that that happens on top of whatever signal we're driving. So there might be some entrainment going on, an entrainment effect. But if you do frequency tagging, the first thing that you see is the sensory-driven response, and everything else comes on top. You cannot just say that this is just entrained alpha. But like I said, my opinion. So I guess the evidence is still out, and there are different perspectives on how you can actually prove that. Good. So final conclusion slides. When we look at frequency tagging, first part has a long history in cognitive neuroscience and unique advantages over other techniques. There is this drastic increase as more people get interested in using frequency tagging. Of course, there's also an increase in the methodological finesse. For example, this spatial filtering approach that we've talked about. And then we need to think about what we actually look at when we drive frequency tagged responses in the brain. And yeah, under which circumstances it is actually possible to talk about entrainment as the thing that we are looking at. Maybe as a final point, I hope that this gives you an idea of how versatile this technique is. And I still find that it is kind of like underused. Maybe what Noor is going to talk about in a few minutes is going to change that because it's quite exciting. It's one of the new approaches, and I think we will also see an increase in applications that use one or the other method to do frequency tagging. That's me done. So I just wanted to thank you for the invitation, and thank you for your attention. And yeah, also huge thanks to everyone who was involved in any of the research that I presented that I was involved in, which is too many people to show there. If you're interested in what we're doing next, this is the lab that I'm working with at the University of Dundee now. And we also have this Scottish EU Critical Oscillations Network going. If you're interested in oscillations, then let us know if you're interested in hearing more about this. Yeah, and I think that's me done. If you have any questions, you can also send them here and get in touch via email or at the coffee break. Thank you.